Hello, everyone. I'm Kelly Eversole, the Executive Director of the International Wheat Genome Sequencing Consortium. And I'm joined today by Jason Fiedler, who will be talking with us about multi-species genome-wide genotyping platform that he's using. And I just want to give it a few more minutes. We seem to have some additional people joining, so I'm going to just pause just for a couple of seconds here. All right, so it looks like everyone is in. Before we get started with Jason's presentation, I'd like to just give you a quick overview of the IWGSC. Most of you are familiar with it, but in case you are not, you'll get some ideas. So we are an international nonprofit consortium, a registered charity here, research institute and charity here in the United States. And we have over 3,400 members, over 900 institutes and companies that are engaged with us in 72 countries. And this would, none of this would be possible, our webinars or our workshops or our coordination activities related to genome resources, without the help of our sponsors. So just want to take a moment and thank Arbor Biosciences, BASF, CIMMYT, Florimond Desprez, NRAE, Illumina, Kansas Wheat, RAGT, and Syngenta. And this particular webinar is sponsored by Illumina, and so we appreciate them arranging for Jason to come. So our vision is, after the reference sequence was published in 2018, has been to develop other tools that can be used to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity, and that has included an additional revised version of the reference sequence, as well as updated annotations. I think we've revised the annotation several times to accommodate for manually curated and functional annotation. So we've been very active in those regards, and we're continuing to work to ensure we have the kind of tools and push towards the kind of tools that are available to many of the other cereals, such as maize. So just to give you a quick overview of your dashboard, you have already on your dashboard a place for you to ask questions. You can do that throughout the presentation, but we will respond to the questions at the end. And depending on how many we get, your question may not be asked live, but Jason has agreed to, in fact, respond to all of those questions in writing, and so we'll have them have a link to them and to the answers along with the video, both on our YouTube and then channel, and then also on our IWGSC website. If you have questions at any point regarding something going wrong, you can put something in the chat, and you please monitor that chat panel, because if there is a reference to a paper, we tend to put a link, and so you can copy that link right off of the chat. Already you can download both Jason's paper as well as or PowerPoint as well as my introduction, so that you can have them in PDF format. And the webinar will be recorded and will be posted on the YouTube channel, and you can subscribe to the channel so that you make sure you never miss a new upload. So with that, I want to welcome Jason Fiedler, who is a research geneticist at USDA ARS in Fargo, North Dakota. And he's going to be talking to us today about a multi-species, low-cost, genome-wide genotyping platform to support molecular breeding in small grains. Thank you very much, Jason, for being here today, and thank you to all who are participating in our live webinar. Now it should go to, here we go. Yep.
All right, well, thank you, Kelly, for that. Let me move this over. All right, So first of all, thank you, Kelly, for that introduction, and to the other organizers at the IWGC and to Illumina for inviting me to speak today. I'm excited to be here to share this project that we've been working on for the last 18 months or so, and really quite vigorously in the last 6 months. And so I think this is something that's going to be very useful for the small grains community. So for anyone out there that isn't aware, ARS, or the Agricultural Research Service, is the in-house research agency within the U.S. Department of Ag. There's about 7,000 employees, and we're spread all throughout the United States. And our mission is to really use science to overcome problems, typically with a regional focus. And the problem that we are addressing today with this work is that breeding is hard. A breeder is tasked with making progress in multiple crop characteristics or attributes, all simultaneously within a limited amount of space, people, resources, and these new lines that come out of the program need to balance yield, quality, resistance to disease and pests, all while being attractive producers and to the consumers. Given that it can take up to 10 years to develop a new small grain cultivar with conventional methods like phenotypic selection, how do we know that crosses or selections made today are going to be useful in an environment that could be quite different in a decade? Luckily, genomics and molecular breeding can help assist in making this process quite a bit faster. So in 2004, the USDA ARS initiated the small grains genotyping labs to support genomic researchers and to help deploy this genomic information into the breeding programs, The Fargo lab here is located right in the middle of hard red spring and durum wheat up here in the United States, up in the upper Midwest. The other 3 labs that were initiated are kind of located near the growing regions of the predominantly winter market classes. We generally focus on kind of supporting our regional communities, but we do have quite a bit of overlap. Additionally, the Fargo lab also supports the yolk community in the US and the sort of southern Canada, and the barley community along with the Pullman lab in Washington. So there is an amazing amount of genetics and genomics work occurring in small grains, and this body of knowledge I feel like is growing every single day. I was just at PAG last week, and it was packed with new information that could potentially be used to advance crops. And one of the main missions of these USGG labs is to ensure that all this information can be transferred into a breeder's hands so that they can use it to apply to make better selections and eventually lead to better varieties that benefits everybody. So how do we actually transfer this knowledge into the program? So here we have a typical breeding scheme for an inbred small grains crop like wheat. Crosses are made and segregating families are kind of quickly inbred and expanded to homogeneous lines that can then be evaluated in more of a field setting. Selections at this stage are made, and then lines are advanced to more expensive multi-location year trials, and then the most elite lines go into even more expensive testing, such as actually baking loaves of bread. And one of the worst things that can happen is that you have a line that makes it through this entire process because it looks really good, it gets to the end and then makes a crummy little bitty loaf of bread. So for the last 19 years, the USGG labs have been collaborating with all of our breeder partners to screen germplasm with diagnostic markers to predict traits. A common place that we do this is kind of right before they go out into the field. So then when the breeder kind of walks these preliminary trials, they have some additional information that they can use to be assured that what they pick here will be more likely to be high quality at the end. So now that's been a great system for large effect QTLs with easy to use diagnostic markers. However, for more difficult traits like yield or like some diseases like fusarium head blight, we need to move towards a little bit of a different approach. So with genomic selection, you can combine genome-wide markers and statistical modeling in a training population to predict performance. This has been shown to work pretty well for quantitative recalcitrant traits that don't have a good single marker, and it's been shown to increase genetic gain in a given program. Genomic selection, or GS, is commonly used right before these yield trial stages. And for most folks that use the Fargo lab, that's about 500 to 4,000 lines. So finally now we've come to a second problem that is really the main motivator for our recent efforts for this array. Efficient implementation of GS really requires, or at least greatly benefits from, a low-cost genome-wide platform. Ideally one that is reproducible or durable. So if we go back to our breeding scheme here, and now instead of running diagnostic markers, we start running genome-wide markers on 500 to 4,000 lines, so then they can have their predicted performance in the field. Another way to look at this is with dollar symbols. So now breeders only have so many of these yellow dollar symbols to spread around, and decreasing this cost of genotyping can either increase the number of lines that can be predicted to really push the genetic gain in the program, or it allows the smaller programs to actually start using this to advance their lines. So for the last 5 years, the 4 genotyping labs have all been developing various low-cost platforms that target several thousand SNPs to support GS in these roles. Now, for the most part, these have been targeted sequencing approaches that really take advantage of the incredibly low per sample cost of sequencing when you use these really large Illumina instruments and high multiplexing. However, in Fargo, we've decided to take an orthologous approach and develop a custom multi-species Illumina array. And we've done this for a few reasons. One is we just simply have a lot of experience and expertise with these arrays, having run all of the iterations of the small ag arrays for the last 15 years. During that time, we've kind of amassed a large amount of specialized equipment to process these things quickly, and we have a very good feel of how this assay works. We found that the Illumina array assays are highly durable with rapid processing times of either 3 or 2 days to get to ready-to-use data. We also find that the assays are really robust, allowing for a wide range of DNA quantities and qualities. This is in contrast to sequencing-based platforms that can be a bit more finicky about the DNA quality, and then it requires some bioinformatics processing on that back end to get at some usable data. And for us, this equates to lower risk of getting the data back on time, especially if we start getting large amounts of projects all at once in kind of our fall busy season. Another reason we like these arrays is that we routinely work with 3 crops already, and we were kind of inspired by the Agricultural Victoria AgriBio wheat barley 40K array. So in 2001, Illumina put us in contact with Dr. Kijan Song at the USDA ARS in Beltsville, who also had aspirations for a low-cost array in soybeans. So we decided to join forces to combine the purchasing power of the 4 different crop communities to get that per sample price of the array as low as possible without a huge initial commitment. So at that point, the USDA soy wheat oat bar 3K was initiated. And so for the rest of the talk here, we're going to go over how I came up with these SNPs, a few technical validation points, and then I'm going to go and answer 4 questions for you. Do the array SNPs match the source SNPs? Can we use imputation to now increase the number of SNPs or upgrade them, or to aid in platform harmonization, like I like to call? Can we use dual hybridization to decrease that cost? And is this array actually any good? Is it useful? And I'll be referring it to as the USDA 3K or just the 3K from now on. All right, so I took advantage of the fact that the Fargo lab has already amassed a large amount of genotyping data, especially for these arrays. So I went into our records and I pulled out all the relevant breeding material and put together an integrated file for SNP picking. I was able to find over 15,000 breeding lines in barley that have the 50K or the old 9K array information. I was able to find about 7,000 breeding lines in oat that had the 6K, array information, and I supplemented that with about 1,000 lines with GBS data. I was also given access to a proprietary Illumina array that PepsiCo had made, so that was really nice. Now in wheat, I wasn't particularly happy with the kind of the US coverage that we had in our 90K and 9K data. So I took advantage of a high density exome capture sequencing initiative that was just finishing up as I started in my role here at the end of 2018. So here the 4 genotyping labs had submit representative samples of their growing region, and they were all sequenced at Kansas State University and Edward Rakanov's group. Brian Ward, who was then a postdoc at the USDA at the time, then put together all this information and provided a really nice integrated data set that I could pull from. So my goal here is to identify 3,000 SNPs in each crop that were evenly spaced along the genome and were maximally useful in as many different market classes, regions, individual programs as possible. So for each crop here, I performed 7 steps. First, we ran cluster analysis on the integrated data to kind of replicate what an individual program or market class cluster might look like. An example of this is just in wheat, where PC1 kind of separates out the growth habits, and the subsequent PCAs kind of separate out the individual market classes, growing regions, or individual program, kind of a proxy for a program. I then interpolated the genetic position from the consensus maps that were available, and really the only way that this was possible was having high quality reference genomes and good quality alignments of all these SNPs. I then binned the genome into either half centimorgan or 1 centimorgan regions, and we picked 1 SNP per bin based on several criteria, such as technical performance of the SNP, overall minor allele frequency, and then the number of clusters in which the SNP had an MAF of more than 0.5 or 0.05. So I really wanted to have SNPs that were not fixed in these individual clusters. After plotting this all out, I kind of found these huge physical regions that had big gaps, and so I kind of filled those in with some more markers. And then I asked the community to sort of send me everything that they were using for marker-assisted selection at the time, and those were all added in as well. Finally, after getting all the probes back from Illumina, I swapped out as many as possible that hit in more than one place. And so what I mean by that is, if we're developing a 50 base pair probe for a given SNP along a genome, we really want to make sure it just hits one place. We don't want it hitting the genome multiple times. We don't want it hitting the other species genomes, and we don't want it hitting the subgenomes multiple times in the case of the polyploids. In barley, we ended up with 3,089 SNPs that were nice and evenly spaced along this genetic position map. And as expected, these SNPs are all clustered at the distal ends of the physical maps. Additionally, 208 of these are either diagnostic for a trait or at least annotated to be linked to a trait. So that was nice to have. In oat, we were 11 shy of 3,000. And again, they're fairly evenly spaced along the genetic map that we had, with clusterings kind of at the end of the physical map, in this case, the PepsiCo genome version 2. The oat content contains 940 markers from that 6K platform, almost 1,100 markers from GBS, and then the rest were filled in with the PepsiCo marker array markers. And of these, 91 are known to either be diagnostic for a trait or, again, linked to something, end-use quality, yield, a lot of crown rust markers on here. We got the most markers with the same nice even spacing along the genetic map, and again, clustering towards the end, the distal regions of the chromosome as you expected. And many of the diagnostic markers that were added here were actually submitted by all the USGG labs. And so these are a lot of markers that are routinely run for the breeders and for collaborators. All right, so in June 2022, we received our first shipment of the arrays, and we started running our validation sets, which consisted of representative lines that were submitted by our breeder and research collaborators. If you aren't familiar with how these arrays work, they generate alleles as fluorescent values that need to be clustered to obtain the actual culls. So sometimes the robot or the algorithm gets it wrong, and you need to actually manually get in there and move those clusters around to make sure that it's accurately representing the data that it's given. And so I had to do this with about half the SNPs that were on this array. But now we have the work done, we have improved cluster positions that should be better for all of our outgoing projects. Additionally, we built in some automated conversion software so that we can directly provide these standard genotyping files for everyone who gets this data. So here's kind of what a really nice array marker looks like. You've got the A cluster here at the 0 intercept, and then the B cluster over here at the 1 intercept. However, when you look at 9,000 SNPs, especially in the polyploids, you're going to see a little bit of everything. So many times these clusters are just a little bit compressed to varying degrees. Other times we see some null alleles that we've tried to annotate the best we could in these sets. In the VCF or HapDap, these come up as gaps. And sometimes you just see ugly ones, and you do our best to pick some relevant polymorphism out of there, but these really need to be looked at for essentially every germplasm set that you have. Now, to get a more comprehensive kind of look at how all these SNPs behave, we can measure the distance between this A and this B cluster, and we can visualize the distances as distributions for this entire set. So in barley, most of these distances are near 1 because these SNPs are all really nice. This makes clustering really easy, and you really don't have to do a lot of manual intervention in barley germplasm. It's a little bit of a different story in the polyploids, where you have about half the SNPs are, pretty nice looking. They cluster with a distance of 1, but then you've got kind of this broad peak that kind of centered around 0.5. And for the most part, these SNPs are okay, but they do take a little more careful curation to make sure that the data is actually represented properly. Just like all the other arrays, the call rates on these are excellent for all the SNPs. Plotted here are the density graphs of missingness on a taxa or line basis. So using just 5% missing data as a cutoff, there's really only a handful of lines that fall above that threshold. And if you go a little farther, there's really none that actually have a significant amount of missing data in these 3K SNPs. Entire sample failures do occur occasionally, but it's usually caused by some mishap during processing, and so they don't even make it to the output genotype file anyway. So linkage disequilibrium, or LD, wasn't really something that I implicitly selected for when I was picking these SNPs, but I wanted to see what it looked like in my selected SNPs in these validation sets, which are fairly broad. What we have here are sort of density plots of this R-square metric of LD collected on pairwise SNPs within either a 10 SNP window or just for adjacent SNPs. So if we use 0.2, an R-squared of 0.2 as kind of an arbitrary cutoff, then the values here are the kind of the fraction of all the pairwise interactions that are in LD or are tagging the same haplotype block. We see kind of a moderate level of LD in these SNPs, and I guess the way that I kind of interpret these plots for, like say in barley, for any given marker, there's like a 56% chance that an adjacent marker is hitting that same haplotype block. And obviously, if you go out to 10 SNPs, that percentage goes down. And then as your genome gets bigger, that percentage also goes down. But you do see some redundancy, but generally, I think we did a pretty good job picking non-redundant SNPs here. So since I want this array to be as useful as possible for as many breeding programs as possible, I wanted to check the distribution of the minor allele frequencies within each program individually. So this is visualized here with violin plots, and the values are indicated here are just the number of SNPs that have an MAF greater than 5%. So it's nice to see that every individual program in barley has at least 2,000 of these non-fixed markers. We get the same results in oat with every individual program that we tested having at least 2,000 of these non-fixed markers. So that's great. Finally, in wheat, again, we see the same thing. Most programs have over 2,000 markers that they can work with, except here, the NDSU Durham program had only a few, and we'll need to investigate a little bit to see how we might be able to address this for them. All right, so let's now move on to our questions. So do the USDA 3K SNPs actually match the source? So for review here, I've got a graphical representation of where these SNPs came from. In barley, the 3K was an entire subset of the 50K. In oat, the 3K was a combination of the 6K and GBS. And wheat, the 3K was almost entirely from these exome capture data set, but the 90K and the GBS is also commonly used in wheat, and you'll see why I include this later. So let's start with oats. I like to use the concordance metric to evaluate how well two genotype data sets match each other, and this is simply just a fraction of matches between two taxa, excluding any missing data points. To visualize, we plot the concordance here on the y-axis by the number of SNPs counted on the x-axis, and this is done twice. We get the blue points here when we exclude heterozygous calls, or the gold points when we leave them in. So here we're comparing the 940 SNPs on the 3K that we took from the 6K to the original source 6K alleles in a set of about 600 breeding lines. Now, immediately, we can see that we have two clusters arise, where the maximum concordance occurs in the blue cluster without the hets. We have more SNPs now if we include those hets, but at a dramatic decrease in our accuracy or concordance. So I think most people probably would trade in these additional SNPs to get higher quality data. And just for some foreshadowing, this is essentially the same behavior that we obtain for every single data set that we have checked. So when we compare the 3K array calls now in oats with the source GBS calls, we get the same results as before. We get about 98% median taxa accuracy in this high quality set of about 500 lines from South Dakota State University. In barley, the graphic is a little hard to see since most of these taxa have 100% concordance between the 3K versus that 50K allele call. And that makes sense because all of these are array SNPs called with very similar cluster positions because these barley SNPs are really, really nice. And finally, in wheat, we see the same results of really nice concordance between the 3K SNPs and the source exome capture SNPs, albeit in a fairly small set of hard red spring wheat cultivars. So it's reasonable to expect that a few of these SNPs are not going to perform well. So you can also look at this concordance on a site basis, and that enables us to remove some of the poor performers if we want to. So here, if we drop 10% of the worst SNPs, our accuracy goes up by a point. If we drop the 700 worst, it goes up to 99% accuracy. This kind of phenomenon also works for all the other crop data sets that we played with too. All right, so now that we're assured that the 3K markers match the source, can we use imputation to upgrade these markers, upgrade these SNPs? Again, we have the graphical representation here, and can we utilize the 3K information of a line plus some reference panel of related lines that contain the calls from both platforms to impute the higher density set? So 3K to 6K, 3K to GBS, 3K to all of these. So starting now in barley, I took a set of 182 barley lines from the University of Minnesota breeding program that were genotyped as part of our 3K validation set. These 182 came out of a very large 6,000 individual strong 50K data set reference panel. So I merged these two together after removing the overlapped lines from the reference, and then we use Beagle to impute the missing 50K data in those 182 lines. I then check the concordance of these imputed markers against the real thing, and then we visualize it in the same way as we did before, except we've dropped the gold visualization, the gold hets, and then we've got red here. I think the results speak for themselves. With almost 100% median accuracy, we are able to upgrade these 3K markers in these 182 lines to about 15,000 non-fixed markers from the 50K. All right, using a similar scheme, we get equivalent results in oat, where we use the 3K markers to impute the 6K in a set of about 500 breeding lines. There are a couple of markers here, or a couple of lines here that fall off, and this is likely due to either poor relatedness of these lines with that reference panel, or potentially just mislabeling of that line when it got put in here. Now, we've now we've only upgraded this set to about 3,000 markers because the 6K array inote actually only contains about 4,300 markers, of which 1,000 of them are on the 3K, and so many of those were just excluded. So really this was just kind of the maximum that we were able to get. All right. Additionally, we can get similar results when we use the 3K markers to impute about 12,000 GBS SNPs in this set of 500 lines from South Dakota State University. Finally, in wheat, I ran several tests, and I'm just summarizing kind of the average values that we get here. With this same set of 100 exome capture lines, I'm able to upgrade the 3K markers to about 88,000 exome capture markers. And then if you drop those spores performing 8,000, your accuracy bumps up to 98%. I've also used the 3K markers in two different breeding sets to impute GBS, and we're getting about 95% accuracy when we do that. And then finally, we can impute the 90K array markers as well, again, in kind of a small set of hard red spring wheat from North Dakota State. Initially, you end up with kind of a small or kind of a low initial concordance of 93, but if you drop those low performing 5,000 markers, you can bump it up to 98 and be really more assured that what you have here is really quite accurate. So now another way that we can impute genotyping data is by using something called a practical haplotype graph, or a PHG. And this is a framework software that's been developed by the Buckler Group in Ithaca, New York, or Cornell. There's been several manuscripts on the PHG now, with the latest one being published last year by Peter. Essentially, it's a database that stores genome haplotype information in a graph. It's got a number of features, but one of them is that you can submit genotyping data, like the 3K SNP information, and it will impute the remaining SNPs on the DB. Katie Jordan at the Manhattan ARS location has been leading the development of a wheat PHG using that US germplasm that was submitted exome capture sequencing. And she's just finished one that contains about 2.5 million SNPs. A postdoc in my lab was also put one together that just contains the hard red spring wheat as well. And depending on the specific options or the tweaks, depending on the data you give it, we can get up to 96% concordance on a subset of about 300,000 non-fixed sites. This database now has been deposited in the Tritici Toolbox, or T3, and this imputation feature can actually be accessed by anyone that wants to submit their data to T3. So please reach out to Clay Burkett at T3 if you want to have more information. So on to question 3. With some of the initial per sample, so with the initial per sample price of the current array of $14 being a bit too high for some, we wanted to see if dual hybridization or dual hyb would work. Again, this was a nice feature of the AgriBio wheat barley 40K, and it's described in their manuscript that came out in 2021. A dual hyb is really simple. You just mix two different crops together and then run them as a single sample on the array, and then you just extract out the relevant data for each crop at the end. Now, unfortunately, we've only had a chance to complete one test with an oat barley mixture, and the results are really promising. Here is the same concordance plot that we've been looking at before, where we're comparing the barley calls in the dual versus the single mode in 4 plates of lines from the 3Ks, from the validation sets. In this case, we got green now to signify that the hets were excluded from analysis. And pretty happy about it. Essentially, we're capturing all of those SNPs back when we're in dual mode. We do see a bit more missing data in the dual mode, and I think that we can address this with some better scripts for calling for clustering and calling allele calls. On the outside of the equation, we obtain, again, very similar results where we're really able to capture the majority of those SNPs in dual mode. One thing that you can notice here is that as the missing data increases, your accuracy kind of falls off. And so we're going to try to use this inverse relationship in the future when we run dual mode to kind of predict accuracy. So then we can tell essentially the customer to expect a certain level, a certain accuracy at a certain missing data rate for oats. So dual hybe tests and wheat are ongoing. Actually, I think that these arrays might be run actually today. In the meantime, a few years ago, we did do dual hybe with the 40K array. We did get fairly good concordance using all the markers, 92 and 94 for barley and for wheat respectively. I'm sure if we go back into this data set and kind of remove those poor performers, we can bump this up to above 98, 99% if we want to. So with this data at hand, I'm really optimistic that dual hybe with our 3K set is going to work pretty well. All right, so for our last question, is this array actually useful? So I see really three main applications for this low-cost, high-throughput array like this. One is for just basic genetic studies where you really only need to identify relationships between individuals and just get population metrics. The International Oat Nursery is going to start doing this with the 3K array now that the 6K array note has been retired. Secondly, I think this is a really great platform for fingerprinting, identifying lines, or identifying impurities in a sample. And I know I'm not the only one that goes out to the field and aggressively rogues out off types to keep that plot genetically pure. And we started doing this internally when we're really not sure about a plot if it's pretty heterogeneous. And we definitely do it when we receive lines, especially check lines from collaborators now to really check purity. Finally, I think this will be really useful for genomic selection, but it depends on the performance of the models developed with this array, right? So I've been lucky enough to have some really great collaborators at the University of Minnesota that have already been implementing genomic selection in their programs. So when I put together my 3K panel back last year, I asked Kevin Smith and Karen Bubin if they'd be willing to run their pipeline with the 3K subset of the 50K that they already had. So not new data, just subsetting their 50K that they already had. Karen happily obliged, and she sent back this really promising data using cross-validation as our metric to assess predictive ability. The models built with the 3K in maroon really performed equivalently to all the models built with 50K. And for clarity, I'm only showing 6 traits here, but they sent me like 24 of them, and they pretty much all look the same. Additionally, Kevin Smith also happens to breed oat, and he has been running genomic selection with the 6K array data. So again, Karen also sent back the accuracy metrics of the models built with the 6K and models built with 446 SNPs that overlapped with what I had picked for the 3K. Okay, so what we're seeing here with this 3K model in maroon is that they're about 6% less accurate than those full models that they're using. And I think that this difference is entirely due to this low number of SNPs. We just gave them new 3K data on a big subsample or a big sample actually in dual mode last month, and I'm confident that the predictive abilities are going to be sufficient when all those SNPs are actually able to be used. All right, finally, Jim Anderson is the spring wheat breeder at University of Minnesota, and we ran about 800 breeding lines through the 3K, and Emily Connolly, his technician, was able to put together genomic selection models with that same set of lines using the 3K data and the GBS data. So 3K is in maroon, the GBS data is in gold. Again, we get really nice equivalent models, equivalent predictive abilities. So this gives me a lot of confidence that this array is going to be useful for a lot of other people as well. So in conclusion, I'm really quite happy about how everything's worked out so far. It operates just like all the other arrays, and the probes have done a really nice job capturing the true source SNP. With imputation, we can increase the number of SNPs, and this is going to be useful to avoid re-genotyping training populations and kind of aid in this platform harmonization thing. The wheat PhD is live now, and I suggest that everyone with some wheat genotype data, you go to T3 right now, you ask for Clay, tell him Jason sent you, and tell him you want to try out the PhD that he's got. Dual hybridization also works really nicely in at least an oat barley mixture, and I'm pretty optimistic that our forthcoming wheat tests are going to work pretty well too. Once that's complete, we're going to try doing tri-hybridization with all 3 small grains next. Finally, the intended application for this is really to support genomic selection, and I think we have really nice initial results to suggest that this is going to work for a number of people moving forward. All right, just to wrap things up, I need to acknowledge a few people for their help on this project. So from the USDA, Mary is our longtime service technician. She is great. She handles all the service work in the lab, and she's runs all this array data. So big thanks to Mary to getting this all out in a very timely fashion. Dr. Kijan Song for essentially collaborating together to help combine these disparate communities. And then Gina Brown, Gadiri, Devin C, Gui Hai Bai, the other kind of leaders of the other genotyping labs for collaborations, providing data, providing their marker-assisted selection CASP assays so that I can put them on this array. They've been really helpful. Bakash was the postdoc in my group that worked on our little PhD, and then Katie Jordan for putting together the PhD for all of the US wheat. Clay Burkick again for kind of implementing, deploying this PhD into more of a user-friendly interface. And then also Kathy Kos for providing a lot of markers for like crown rust resistance in oats. Outside of the USDA, we had a lot of collaboration as well. Mandy Waters at PepsiCo was instrumental in sort of providing all of those PepsiCo array snips for us to do filling for the oat content. Dr. Akinov at KSU did all the sequencing for the wheat data that we used to pick. Brian Ward, who's now at Land O'Lakes, did a really great job putting that all together. And then really just kind of generally the small grains community, we've had a number, when I was developing this, we had a lot of conversations with a lot of the customers, a lot of the people that we work with, and we really wanted to make sure that what I was developing was going to be useful, and I got a lot of feedback from them, and so that was really nice to make sure that I was on the right track. Specifically, a big shout out to our collaborators at the University of Minnesota for providing that genomic selection data so timely, has been a really great help for me. And then the folks at Illumina kind of help put this all together. Matt and Khalil are now no longer working on this project. Yuting Bai is now our contact for these ag arrays, and then Brenda is our sort of our local person. And then sort of the funders who sort of either explicitly funded this project or implicitly helped by going to Congress and being kind of a great stakeholder to be our advocates. And so with that, I think I will let Kelly come back on, and I'll see if there's any questions out there.
Thank you very much, Jason. Really interesting presentation. And we do have a number of questions that have come in. Just to remind everyone to please put your questions in the Q&A or the question box on the panel that you see from the webinar. All right, the first one is, is the platform based on single copy sequences or genes?
So the arrays were originally designed using exome capture data. And so those were sort of focused on genes. For the exome, yeah, so actually all of the things were based off exome capture at its source. And so they should be targeting more of the genes, which is why we see them more in the distal portions of the physical maps. The only ones that wouldn't would be that 1/3 of the oat content that is just GBS, and GBS is just kind of a random smattering throughout the genome.
Right. So someone asked, do you plan to compare this to corn? And I do think that would be interesting to see if this compares to the results that have been achieved in maize. So do you have any plans for that? Or are you collaborating with anyone working on maize?
Nope, we don't have any collaborations in maize right now, but I think that if anyone out there in the audience wants to work on that together, I'd be happy to do so. Please reach out. I have my, let's see, I've got my email address right here. Please send me an email and I'd be happy to work on that.
So the next question is, when you designed multi-species array, the probes that hit 2 or more species were removed. Can you explain the reasons behind this step?
So they weren't removed de facto. So I did my best to try to limit the number of cross hybridization, but I knew that was going to be a problem because you've got conserved genes that we're hitting. So I on some of them, it was one of these things where I got the probes and then I would run the alignment against the other genomes. I take a look at the hits and the scores of those hits of the probe sequences. And then if I was able to find another SNP in that region that was sort of equivalent, like equivalently useful, I would then remove it. But if it was really only, if it was the only SNP in a region and it just happened to cross hybridize, I just sort of left it and I thought, well, you're going to have competition between the two genomes. Likely the one that's more cognate would work. So I guess it wasn't a hard and fast cutoff, but I did try to limit it if it was a possibility.
All right, are the 3K SNPs available broadly to the community, and if so, where?
Yeah, so I have been providing the list of the SNPs to people just when they ask me. This is all pre-publication. I do have a couple things obviously to still button up here before we get this written up. It'll be published when we get it written, but if you're curious to see what's on here, please again reach out to me and I'd be happy to provide that information.
All right, and so once you do publish, what will be the availability outside of USDA or the wheat cap, for example? And what would be the unit cost for single and dual hybrid? Yeah.
So I wanted to have this array be as useful as possible to as many people as possible. So it is, I don't have any restrictions on who can buy it. So as long as you have the ability or you have a service provider that can run Illumina arrays, you're able to buy it. If you go through a commercial provider, the price might go up a little bit because there needs to be some margins there. If you run it here in Fargo, we get a price of $14 per sample. That's what we initially sort of negotiated, again, with that kind of that low amount of initial commitment. And the way that we work is that we don't really recharge. We don't have an avenue for invoicing. So we generally just have the folks drop ship. So they order the reagents themselves. They come to Fargo. You send your DNA or your tissue or your seed to Fargo, and then we do all the processing and then send the data back. I think that once you start to get outside of the United States, to folks that maybe I don't know, I don't work with regularly, the price might change, especially in different countries. But I think that that can all be a negotiation, and I think we can talk about it. So the way that I'm thinking now for the folks that want to use single or want to use dual, is to kind of work on the logistics of finding matched pairs. And so we'd mentioned Kevin Smith's genomic selection material. Since he already breeds for 2 crops, he just sent us them together, and then we were able to easily just put them together and run dual hyb. We still are kind of working out the logistics and how we might do that as more of a service. But right now, it's really more on a per project basis.
All right, is there plans or flexibility to add new targets for the array over time?
That's a great question, and yes, absolutely. So you never expect these things to work perfect the first time, and you want to have the flexibility to be able to upgrade things. So there's 2 ways that you can do this. You can do it through a new, like a sort of resynthesis of the entire bead pool, and that can probably occur every maybe 3 to 4 years, because you do need to have kind of an initial negotiation about price and all that. I think a easier route is to do something that Illumina calls add-on content, where you just say, I'm gonna we want this 3K array, but let's say there's 1,000 or 500 SNPs that we really want. We want to order add-on content, and so they would synthesize just that little small bead pool, and then it would be like a surcharge on top of it, maybe $1 or maybe $2. And that's the way I kind of see going forward is once we get kind of all of our things wrapped up, everyone gets a chance to kind of see how it works. Then we'll start talking about how we can add things, and also add new things that we want to add, right? So like, again, we're learning new things every day. We're cloning new genes every day, and so I want to make sure that we can continue to improve this array so it's as relevant to what we're breeding for right now as possible.
So just to dig in a little bit more on the lines that were selected, were any of those elite lines that are owned by companies, or were they all lines in the public germplasm, either at ARS or at the universities?
So, most of the folks that we work with are the public breeders. So the folks that are at academic institutions and then people within the USDA ARS. And so I didn't really give any restrictions on what they could send me. I just said, send us enough lines that when you run it, you'll be able to really dig in and know that it's going to work in your program. And so we didn't have any companies come to us directly. I believe that some companies have started running it internally, at least as service providers, and so I don't have access to that data. I don't know how well that works. If there are any company folks out there that want to give some feedback on it, I'm happy to hear that. So I think that my goal is to try to improve crops in general, and so that can happen in public spaces as well as private. So yeah, I think the majority of lines were breeding germplasm, things that are kind of in the pipeline of advanced field trials, elite lines. A lot of checks were in there. And we did run the nully tetrasomic lines, and so we do have to kind of dig into this a little bit more to see how well these arrays work.
All right, do you have any plans of putting more than 2 crops on the chip?
So right now there's 4 crops on the chip, and we've tested essentially doing dual hive, and then we'll be testing doing 3 samples together. And actually, we're not averse to doing 4 samples together to see how well it works. I think that as you increase the number of samples you're mixing together, the ratios get a little bit more complicated, right? And you need to make sure you have really good DNA quantification, which sometimes is a little bit tricky. And so it gets more complicated, but I think it's very doable. Again, we're kind of following in the footsteps of the folks in Australia, the agricultural Victoria people. I know that they're running these multi-species arrays a lot, and they're doing, I think, quad mode, and they're having really good success. So, I think that these are all things that we can try out. Additionally, we're not averse to adding more species to this array. So the format that we have right now, I think can handle up to 9 different species. You can have 90,000 SNPs total. So there's a lot of room for this to grow. So if there is community out there that wants to have sort of content on here, I think that's a conversation that we can have about how that would work out.
All right, well, the next question is whether you think a prediction accuracy of 0.4 to 0.6 is really sufficient for genomic selection, in particular from wheat.
Yeah, that's a tricky one, right? So if we go back to those arrays, let me just go back a couple slides here. Yeah, right here. So I think so we do get some pretty high variances on these cross-validation metrics, and that's obviously not ideal. I think as we get more lines put into these models, I think that variance will go down. 800 lines is I don't think would be sufficient for really using it. I think it's okay for initial, but it's something where I think the more you add, the more location data you add, I think that, again, it makes those models a little more adapted. So yeah, models with high variance like that, you have to kind of take with a grain of salt.
So have you, at least in what people will get and be able to see, it will have the location data on there as well?
I'm sorry, I don't understand.
In terms of tying it into, I'm actually thinking of a broader genotype to phenotype, looking at including that might include weather. You might be able to layer weather on top of it if you know exactly where the field trials were held, for example.
Yeah, that's getting into the whole just better improving prediction models in general, right? And so, I mean, this small set of SNPs is one part of that, but yeah, I mean, including all of the trials in as many places as possible, plus all the weather data. Yeah, that's going to be how we push this forward to get higher accuracy. Because right now, I think a lot of the models are being used for triage and less for selection, and so. I think until we can kind of get better at this, it's going to be a tool. It won't be the sole thing that our breeders are using.
All right. Are there any modifications required on iScan to be able to run the chips?
Well, that's a good question too. Yeah, I think you have to have an upgraded computer, which I think is just part of their maintenance that they've done in the lab a couple of years ago. When you're running the higher throughput arrays. So in the arrays, there's HTS, which is kind of the standard. It has 24 samples per chip. That's what this looks like. This is 24 samples per chip. When you move to the next format up, called XT, that's 96 samples per glass slide. And so they really recommend getting their TCAN robot that they programmed to do automatic loading. And that makes life a lot easier. The TCAN robot can also do all the staining automatically. And so we've really enjoyed having that. For a long time, Mary was running all of this HTS by hand, and she was good at it, but it did take a lot of time. And so having that robot really adds to quality of life for your service technicians.
So if you were able to use the entire array for wheat, how much haplotype coverage of the genome would be gained?
So that's a good question too. So if you look at like an individual, okay, so there's two things here. Within an individual breeding program or maybe a market class, your LD blocks are quite large, right? So if you look at, say, hard red spring wheat coming out of just the North Dakota State University program, your LD blocks are somewhere between 5 and 10 centimorgans, right? So we're definitely hitting all those blocks multiple times within an individual program. The trick is I'm trying to hit as many individual program blocks in as many programs as possible, right? So you need to have sort of redundancy built in, so then you can hit all the blocks in North Dakota State, all the blocks in University of Minnesota, all the blocks in North Dakota State University, all the blocks in Washington State University. And so that's the kind of the I guess the balancing act that I'm with. As far as like as many as possible, that's something that I haven't really dug into, and I'm sure we could get a lot of them, but it wasn't really the intent of this array to try to hit everything. It was really to be really focused on what the breeders needed.
So do you think that these data can actually be used to develop a Thermo Fisher Axiom chip, for example?
Yeah, so Thermo Fisher has their Axiom array, which we don't run. We don't have any equipment for that, but the SNPs are all the same, right? You just give them the SNP location, the sequence surrounding it, and then it just goes into their probe development software. So yeah, I mean, absolutely could be run as an Axiom. And again, I don't have any proprietary. I'm not holding on to any of this information, right? I want this information out there. So if someone, especially in Europe, where I know they run a lot of the Axiom arrays, wanted to convert this into Axiom, that'd be fine by me.
All right, great presentation, Jason. I think, and there are other questions that came in, but we have about 1 minute left. Is based on the questions, is there anything else you would like to kind of throw out for the audience?
I guess, one of the things is, if you are interested in using this array, please reach out. Again, let me bring up this one again. Please reach out to me. We'll get you connected to the service team. And I really want to see, especially because I've developed this array for, really focus on US germplasm. I don't really have a good sense for how well it works in germplasm outside the United States or in Canada and South America. And so if you're someone sitting there and you want to kind of try it out, please reach out. it's hard to send seed overseas, but you can send DNA, and we can definitely give it a shot as a trial run. So please reach out if you're interested.
Great. Well, thanks again, Jason, for a great presentation, and we all look forward to seeing the publication hopefully later this year. And I want to thank everyone who stayed with us for this great webinar, and we look forward to seeing you on our next webinar. So thank you very much, and have a great day.